evening. We have a treat. Dr Rachel Kruft Wilson, one of our members, is a spider enthusiast. I have to confess to being a bit of a spider enthusiast myself. And so she's been researching the fossil history, really, of spiders, and that's what we're being treated to tonight. So, over to Rachel.
Right. Hi, I know some of you have seen this before. It's got a few minor tweaks to it from previously, but you will recognise an awful lot of it if you have seen it before, so apologies. The talk is basically, I'm going to take you through what we know about the fossil history of spiders, a lot of which is known from amber, and then afterwards I've brought some specimens along for you to have a look at, but I've neglected to bring a light, so you won't be able to see anything down the microscope at all. So you'll have to peer at I've put my hand lens out, but it's really not going to work. You're just going to have to hold them to the light or something.
Torch on camera?
Torch on camera might help. You can give it a go and see if you can see anything. Otherwise, you'll just be looking at pots and going, wow, that's amazing. Right, so the amber spiderglass. Spiders as fossils, and I apologise, I'm going to have to sit down fairly often because my knees are absolutely destroyed and I can't stand up for very long anymore. Right, the spider fossil record is pretty ropey, to be perfectly honest. There aren't hundreds of bones of spiders lying around, mostly because spiders don't have bones. They're fragile, they're soft. They're also not very big, which makes them a little bit harder to find than the average woolly mammoth. And when something like a spider dies, it doesn't take much for the grit and sand that it lands in to wear away the remains of that spider. So there's a lot that gets destroyed before it even gets a chance to be fossilised. They're also going to be just snapped up by the average decomposer, the birds wandering around, oh, snacky spider, yum, yum, and it's gone, which is something that when your woolly mammoth dies, yes, there'll be scavengers, but they won't take the big bones. Whereas with a spider, it's one snap and it's gone. So there's a lot less available to be fossilised in the first place. They also tend to live in environments that are not very prone to fossilisation. So this chap here, for example, is living in a very arid environment. It might live in a burrow, but when it dies, the fact that it isn't damp means that it won't cement around the organism. Other things, let's go on, might live in forested areas where if it dies, it's going to get trampled and destroyed and churned with the mud. And again, there'll be nothing left. So fossil records of spiders, not great. Right, I'm going to introduce you to two words that you may or may not know. The first one is taphonomy, and the second one is diagenesis, which is fabulous. Just hold the thought. Sorry? Quick. Okay. Right. Taphonomic processes are things that change an organism after it dies. Hang on. What have I got? There we go. Diagenetic processes are the actual replacement of the minerals in the organism to produce a fossil. So more taphonomic processes might be things like parts of the organism become pulled apart by scavengers or bleached in the sun, things like that. They could be swept away by wind or water. There was a fox over at over in Far Forest, and it died in the woodland there, and it was in a kind of remote bit of the woodland next to a stream, but I used to go past it quite often, and I watched it slowly over the course of about 4 months. This thing slowly disintegrated and slid down into the water until only the skull was left. The rest of it sort of just got washed away. So the skull was the heaviest bit, and that was left. So if I was digging up this fossil later, I might only find part of that organism because the rest has gone somewhere else. Right. What we need when we want fossils is to actually bury them very, very rapidly somewhere where there's no oxygen, because the oxygen allows things to decompose it, allows the bacteria to turn it into slush. So we want to bury it rapidly, and rapidly with no oxygen. Lake beds are ideal, but unfortunately there aren't very many spiders down there in the lake beds. But they do sometimes turn up in them, just sometimes. There are some water spiders, they do exist. This chap is called, bonus points if you can pronounce that one, Argioneta aquatica, the diving bell spider. And this thing's pretty awesome actually. It wraps itself, or its abdomen, its back end, in a big bubble. You can see this big bubble here. And it actually uses that like a big membrane to exchange surface so that the oxygen can diffuse in and the carbon dioxide diffuses out. So it's like this massive external lung that it's got. It's really quite neat. So there's this thing, but the fossils that we do find in lake beds don't tend to have a lot of these in them. They tend to have other things in them. So we think that spiders that turn up in lake bed fossils preserved alongside the groups of aquatic organisms that are down there, possibly fell in, or maybe got washed in during a flooding event. We've had plenty of floods this last couple of weeks, and all the meadows and everything else that got flooded then drained back into the waterways, taking with them things like our spiders. So we think that that's probably what happened. One of the problems for paleobiologists, of course, is that if you're finding spiders in a big assemblage of aquatic things, it's really difficult to try to reconstruct what they were eating and how they were living back in the prehistoric times, because you've basically found them alongside some coral, and they're probably not eating coral. Right. Let me tell you a case study of one particular fossil that was found. This thing, okay, is an Argentinian fossil, which dates from the Carboniferous, so we're talking 300 million years ago or so. It was described by this guy 40 years ago, 45 years ago, and it was found in lake beds, which contained some aquatic stuff and some terrestrial stuff. So he went, yep, that's a spider. Look at the size of it. Now I've measured this. It's about the width of my shoulders. That is a whop-off big spider. Okay. How that got washed in, it must have been one hell of a tsunami that washed that in. So they did a bit more research on this, and it spawned eventually the following headline. Well, thank goodness for that, eh? Okay. It was actually one of these. It was actually a sea scorpion when they finally managed to look more deeply into the rocks, into the embedded parts. And I'll come to how we did that in a minute. They found that it wasn't a spider, but it was still an arachnid of type. It was still related to them, but it was definitely an aquatic beast, not a spider. Right, let's go back to the history of spiders. Before spiders, were these things called trigonotarbids. And these are possibly my favourite organism. Okay, I love a good trigonotarbid. They're fantastic. Okay, they evolved a long time ago, so we're talking 400 million years ago in the early Permian. And these were the first fully terrestrial arachnids, and I'm going to tell you the story of how we know that they are terrestrial as well. And they probably ambushed small invertebrates, but that hasn't changed because spiders still do that now. Okay. Right, what's the difference between a trigonotarbid and a spider? Right, so a couple of words that you might need to know that I'm going to bring up later is that one, which I'm going to use almost interchangeably with abdomen because I can't pronounce it. I end up spitting over people. Opisthothoma. Yes. Right. So I'm going to call that an abdomen. It is basically the back end of a spider. Spiders come in two chunks. The head and thorax, which is called the cephalothorax bit, and the back end, which I'm going to call the abdomen. So this is a trigonotarbid. It's got different lumps for where the eyes sit. It's got a segmented abdomen, and it doesn't have any spinnerets. That's the bottom. That's the basic differences. So it's not quite the same, but it's got quite a lot of similarities. There are still some spiders, these guys. Just notice this is just going off the edge of the... It's okay. But there's an L in front of that. Lephistid spiders that still do have segmented abdomens. And again, we'll come back to those in a minute. Okay. This is one of the spider fossils from the Lapworth Museum that I did some work on a couple of years ago now. And, sorry. Oh, okay. And this is the part and counterpart. So these two fit together on top of one another. And this is actually a trigonotarbid. But from looking at it, there's a lot of material hidden in the rock that we can't see just on the face of it. This is a three-dimensional fossil, but it's all hidden in the matrix. So what we can do is we can CT scan these things. Pardon me. And so we use X-rays and we image the fossils embedded in the rock. It picks up the density between the matrix and the fossil because they are made from different minerals. You remember our diagenesis word some time ago, where we replace the minerals with a different sort. So the densities are different and we can pick those up. And then we can spend a lot of time with software and reconstructions to piece these together to make a three-dimensional model of what we've got here. So, right, scan, yeah, hidden structures. And this is going to twiddle. You can see the gap between where the two halves of the two pieces of rock were put in the scanner, but with a gap between them. So there's quite a lot more hidden in that rock than was visible from the top. And if I was clever enough, I could cement those together. I'm pretty sure I can do that, but I haven't ever actually got around to it. So to get a more three-dimensional thing. You notice that the head is missing on this. We've only really got the front of the spider there and a little part of its abdomen. Spider, it's a trigonotarbid. Okay. So that worked okay. There wasn't a lot in that one, but we did it. We now know what's inside. Okay. Lagerstätte, which you probably know this word. This is a site where there is really, really awesome preservation. So the fossils that come out of Lagerstätte, there's lots of them, and they are beautifully preserved. And there's different ways of Lagerstätte doing that. This one if I can get it going, comes from the Rhynie Chert near Aberdeen. This was the organisms there, and there are a lot of them, are so exquisitely preserved because there was a volcanic hot spring and it invaded this marshy area that was full of creatures and plants and things. And this water, because it was volcanic, it had lots of silica in it. So not only were these things instantly, well, boiled alive, I suppose, preserved very, very rapidly, but they were preserved in something that mineralized them straight away as well. So what we get is that we can even see cells in some of these things. We can see the stomata, the holes in the bottom of leaves. It is absolutely incredible, the preservation in this. And this is a trigonotarbid that came from there. So again, the part and counterpart of the same creature. And if you go to the Lapworth Museum of Geology, they've actually got a 3D model of this on display, so that they've blown it up to about this size, and they've 3D printed it, and it's in one of their display cases. He did say, did I want to borrow it? But I was like, no, I don't want to break it. So I haven't got that. I have got a 3D model of something else that we've done the same to here. This is an ant, which you can have a look at in a bit. That we've done something very similar to and made a 3D model of. Might have to sit down in a minute. Okay. Yes, I told you I would promise to tell you how these things were terrestrial. So what we need to know about these, this is the inside of a modern spider. And modern spiders have these things called book lungs, which are laminated layers of lung tissue. So we can allow air to flow between them for gas exchange. So a lot of marine creatures have book gills, but they stick out from the organism, and book lungs are embedded inside organisms. So something like crabs might have book gills that stick out a little bit, but terrestrial things have them embedded. They are only found in land creatures. They can store air for a few hours, but they can't be used underwater. They can't be sealed off. So they can store air for a little while, but they are definitely a sign that these things are terrestrial. So we went back to our Rhynie Chert trigonotarbid, and we had a look. Well, wasn't me. Some scientists had a look. And okay, I know there's a lot going on in this slide. Start up here. This is what the book lungs of this type of arachnid look like. And this is what the book lungs of this type of arachnid look like. All right, so we're looking for something that is layered. All right, so far so good, I haven't lost anyone. This is our trigonotarbid, preserved in the Rhynie Chert. So we're going to focus in. If we focus in on this bit, we get this picture. And if we focus in on this bit, we get this picture. And here are your laminated book lungs, beautifully preserved in a fossil 400 million years old. That is pretty awesome. I'm pretty impressed with that. And again, they've made a model reconstruction of what these look like. So we know they were terrestrial because they had that particular structure. Right, okay, so we're moving on from my favourite organism. Okay, looking at other fossils and other spiders. So the earliest records of things that might be spiders is this laminated, flat, two-dimensional thing from approximately 390 million years ago. Atacopus means poison head, and I think the Norwegian for spider is still atacop, something like that. I think it's still a word that exists, along with crochet patterns of them, apparently. Christmas is coming, you know what you... All right, so this is one of the earliest ones. It's got this long thing at the back here, which originally, when people pulled this out, they went, that's a spider, look at all those legs, that must be spinnerets at the back there. Awesome, we've got the earliest spider ever, love it. It is not a spider. Yes, it's got this long thing at the back, but these are not the same as spinnerets. It's in a closely related group, which they named, and again, points for pronunciation. Uraranidia? Anybody? Yeah, something like that. Okay. It's there. So what it actually had when they looked closely is that it had some pores underneath the middle of its back end. So not at the back where spinnerets are, but in the middle underneath. And those would have produced silk. So it did make silk, but it just didn't have the organs that could make it into a nice spinny thread. So it was probably using silk to line its burrows or keep its eggs moist. Remember, these are things that have not, in evolutionary terms, been out of the ocean for that long. Their eggs are going to need to be kept damp. They're not necessarily going to have protections on them like chicken eggs do now, with a nice shell that keeps all the moisture in. They might well be drying out. So it could be that they've got behavioural things, and it could be that this silk is forming a protective layer around things. Not a spider. Okay, let's see if we can get a bit closer to spiders. This is our next one. This thing, much, much more recent. Spider cousins come from Myanmar, which was Burmese amber. Now, Burmese amber is a massive issue in paleontology because Burma is such a closed country. Actually, exporting amber from Burma is a huge legal minefield. So a lot of amber was taken from Burma without permits and things. So now, unless you have a permit for the amber that you are looking at, very often you can't get your work published. So if I go back through some of the amber from like the museum collections, there's no permits there. What I look at, I can't publish because a lot of journals just won't look at it in case you've stolen it from Burma, from Myanmar. So it's a total minefield at the moment. I've been working on some stuff. Sorry, I'm going off on a tangent here. I've been working on some stuff from Myanmar that was bought in China, and it was probably legally imported into China and legally imported from China to the UK. However, because there isn't a direct link, I can't go and give a talk at the Paleontology Association in December, which I wanted to do on that, because I don't have the permits. So yeah, they're going to have to sort something out, and I don't know quite how they're going to do that, but it's not, yeah. This one, again, not my work. I think I can do this one. Chimera arachne ingai. Right, lived in the Cretaceous 100 million years ago. This had both a tail, like the Atacopus, and it's got some spinnerets. Look at these little things sticking out here. So it's got both. So it might have evolved from something like Atacopus, and it lived alongside true spiders for a very, very long time. Okay. Right. Right, so in this chap, our spinnerets are right at the very end, just like modern spiders, whereas our Atacopus had silk-making glands in its abdomen, on the bottom of its back end. Now, do you remember I said there's some modern spiders that have segmented bodies, like this one here? And these still exist. There's not very many of them, but they are still around. These have their spinnerets on the underbody instead of at the end. So now we're saying, well, did it have a tail and spinnerets in the middle, or did it not have a tail and spinnerets in the middle, or how did evolution work? This is quite confusing. There's no nice linear explanation for this, because some things have got more than one option. Have I lost everyone? No? No? Good. Okay, because I'm about to. Right. If we draw out an evolutionary tree, I'll try and make this a little simpler in a sec. If we draw out an evolutionary tree, our stripy butt spiders here fall in with all the modern spiders, the tarantulas and the orb weavers, the ones that we know all about. So these are all the modern species down the bottom here. Okay. Other types of arachnids, we've got our vinegaroons, that's these things. We've got the amble piggy, these, whip scorpions. And we've got our modern spiders, which are the three bottom ones. So you can see, well, we've kind of lost a bit at the end there, but we can see all the spiders land together in one place. Okay? And annoyingly, I'm going to apologise now, the next one has got this whole graph the other way up, because that's what I found on the internet. And so you're going to have to turn your head the other way up. Okay. So we've now got whip scorpions and vinegaroons down at the bottom here, and the modern spiders up at the top. So what we've done on this one is I've put the fossils on there for you so that you can see where they fit in to the other groups that we've got. And our dear old trigonotarbids, they reckon, are more closely related to these guys than to spiders, which makes me very sad indeed, because I do like a trigonotarbid and they're not even on the branch that led to spiders. That's sad. Okay, our thing Urorhinidia with the tail, this Atacopus, seems like it was quite a basal spider, very early on the branch to spiders. And the Chimarachne, the one that had both the spinnerets, this one, and the long tail, that's a little bit more modern, but again, it's branched off before we get to all the modern spiders here. So this is our thing with the segmented abdomen. These are all the arachnids. Okay, there's one group missing from here, but that's okay. So these are all our modern spiders up here. Is that all okay? Yeah? Right. What is the oldest spider? This is a case of be careful what you Google. So you Google, what was the oldest spider? And The Guardian, 2018, gave me that headline, which was not what I was looking for at all. True spiders evolved around 370 million years. So we're looking just after the Carboniferous, just after. Yeah, just before even. Carboniferous. One of the first, that's another name for the segmented butt spider. One of the earliest spiders with the segmented abdomen dates to 295 million years and was found in France. France seems to be quite popular for finding things, actually. Yeah, and here is the best picture we've got, and you can see here we've got the abdomen segmented, yeah, quite nicely there. I originally gave this talk to the Tarantula Society, so that's why there's a bit of a focus on tarantulas in places. Okay, so tarantulas are, if we're doing evolution, we've got the stripy butt spiders, then we have things like tarantulas, and then we have the modern garden spiders, and they're kind of in three different groups. So our first tarantula type spider, which is my Gallimorphs fossils, are a little bit more recent, so 250 million years. Again, France, who knows? I don't know why France is so popular. It's not a great picture, is it? Doesn't win any prizes, that one. Oh, I was expecting more. No? Okay, that's all you get. Right. Why should we care about amber? So obviously this is the next bit in the talk that I'm going to do. One of the Myanmar amber that I have been looking at, we actually found a Gallimorph spider in that. So that would have been this Burmese amber that I now can't publish. has got one of the tarantula type spiders in it, which is quite awesome. Okay, why do we care about amber? Well, so far I've been talking about Carboniferous spiders and very, very early on spiders. But sometime around the Cretaceous, plants evolved to make amber. And they started trapping things in the amber and trapping things in the resin that oozes out of damaged trees, which then gets turned into amber. Sorry, how am I doing? And the vast majority of the spiders that we know about have been found in this way. You can see that that's 800, and this is like 10. So massive increase in the amount of data that we can get. But not until, that's this bit here, just after the Cretaceous, that's when the dinosaurs went extinct, that point there. So the vast majority of these are after all the dinosaurs, after that big asteroid came down and did terrible things to us. Most of the amber that we've got is Baltic and Dominican, around this area. Most of the stuff that I've put out here is also Baltic and Dominican. Right, okay, again, I'm going to simplify this. Amber is fossilized tree resin. Part A, this bubble here. Some trees produce resin when they get damaged. I always used to peel bits off the damson trees at home when I was little. It's like oozy, gungy bits of resin. B to D, it's sticky and small things get stuck in it. E to G, E, G, F, the resin gets preserved and buried. And finally, diagenesis, that another one of those great words, we change the minerals inside it so the resin hardens and changes into amber over time. And amber comes in lots and lots of different forms. So first of all, it's going to become copal, which is the darkest form. And although it might still have things trapped in them, we can't see them unless we CT scan them. So given infinite amounts of resources, yes, we'd be CT scanning great lumps of copal and finding out what's inside, but it's quite an expensive job, and it's it's a bit hit and miss. So most of the time what we're going to do is look at the ones that are much, much clearer. So we've got copal, and it's not always transparent. So some of this is called clouded amber, where it's actually quite opaque. And sometimes the amber itself is embedded in other bits of rock, in coal or in limestone. It can be, it doesn't always come as nice blobs of beautifully faceted amber, unfortunately. Okay, lots and lots of stuff. I just want you to look at the yellow stripes up and down here. Okay, we have Teme. This is the oldest at the bottom. This is modern at the very top. I'm going to go over the other side. Slow, slow. Okay. Right. Some ambers were formed in the Triassic. So that's down here when we've still got dinosaurs around. But the fact that we need pines and some other flowering plants that actually make a lot of resin, these had to actually evolve before we could start to get huge amber deposits. So that's when they start to evolve. That says origin of pines, and that says origin of angiosperms, which is flowering plants. So those two coincide with lots and lots of resin deposits all the way up. And then we've got a bit of a gap right at the very top, because it can take a lot of time for resin to turn into amber. So there's not any very, very recent amber fossils. It's going to be copal or gum or other things that we can't really see into very well. So we have a massive batch here, and we have some more up here. And that's where most of our information's going to come from. And I've got a completely misplaced slide right at the end that fills in this little gap with tar pits from the, but I'll just stick it in at the end randomly. So we can get some very recent deposits, but not in amber. Okay. This is what I've been doing for the last, 18 months at the Lapworth Museum. I've been looking at different things enclosed in amber and drawing them with my beautiful drawings. Okay, maybe I'm not going to win any prizes for them, but this is what I can see down the microscope, and this is what I was hoping to let you see down this microscope, but I've forgotten the lamp, so you're not going to see them. But you can have a look through the hand lens and see if you can spot some stuff. Oh, too far. Right. And I've also been working on this Myanmar stuff with Jakob Winter. So these are Myanmar spiders. We found not only the tarantula type one in amber, but we found the first male of a species in amber as well. Possibly this chap, because this one's got really long spinnerets at the end, so we can tell exactly what species it is. Okay, so we can use microscopes, we can CT scan these, and we can even 3D print them, which I told you about that at the Lapworth Museum. This ant, you get bonus prizes if you can find which one of the many many tubes this ant came out of, because this is 3D printed from one of the tubes here, but I don't know which one it is. So your challenge after I finish speaking is to find out which tube it's actually in, the original. So we've we've done, yeah. Okay. Okay, I've also brought along some amber that has problems with it, that makes it more difficult for us to do the job that we want to do. So as amber ages, it can become brittle and crack, and sometimes we get fissures through the amber. Some of them have got lots of air bubbles, and we can have dust and bits of grit in there, bits of plant, and sometimes the organism just dies in such a position that you cannot tell what it is because it's covering the feature that you need to see. Nothing you can do about it. Right, why are there fissures? Well, what we actually have is that some organism will come along and approach the sticky resin, and it becomes trapped on the surface. And then more resin forms over top of it. So we end up with a fissure line where the two resins sat together, where if you're looking at it down a microscope, you can't see the whole thing. You can see that side, or you can see that side, but you can't see it together because the fissure forms an optic break in the plane. So you can't see exactly what's going on. And of course, there's air bubbles and inclusions and all the other things that can happen. And I have brought a couple of badly fissured specimens along, if you can have a look at those, if you want to. Right, taphonomy and diagenesis, our favourite words. Okay, taphonomy. Sometimes spiders that are then stuck in the amber might actually end up pulling their own legs off as they struggle to get free. It's kind of sad, but it was a long time ago, or scavengers can come and pull bits away, or the parts can get worn off. This stuff is a mineralization problem, but it's actually it's got a special name. It's something like vermunglum or something like that. It's called. Please don't quote me on that. It's basically, as this thing started to decompose, some of its innards came out, and then its innards mineralized, and what we've got is this fuzzy mineralization all around the specimen that we then can't see the bits we want to see. So this is a diagenesis problem. And I brought at least one specimen that's very fuzzy. Okay. Right. This is another 3D one that we identified almost. Well, I don't think it is this, but it's something very close to it. So that's this. It's probably within the genus, or at least very close to the genus. So this one worked very well. We looked at it in the amber. We CT scanned it. We 3D printed it. I think, did I bring this one along last time? Okay, can't remember. Did I bring the model along last time? I haven't got it this time. But it worked very well, so we've got a beautiful thing, and we could take it all the way, not quite to species, but we could take it very, very closely. So sometimes it does work out perfectly. Okay. Oh, that must be bringing me to the end. Hang on, I'll come back to this slide. So these are some of the pictures. There's actually a couple of tangled ants down there somewhere. This one's very fissured. Some of them are mineralized. I can't remember. This one's mineralized. So this one's got a lot of fuzz on it. But these are some of the specimens that are down there. And it says, please don't mix up the labels and never let me borrow them again. Photo credits. And this was my random slide at the end. There was that slither just at the very top where there's no amber. But we can get things from about 10,000 years ago, 20,000 years ago, that are preserved in tar. And this is a jumping spider from the Le Bray tar pits. And you can see it's a jumping spider because it's got these massive, great big headlamp eyes that they have bifocal vision, so they can actually judge distances to jump at things. It's kind of awesome. Right, let's go back and let you have a look at what you like over there and have a cup of tea. So.
Thank you very much, Rachel. Would you be prepared to take one or two questions?
Oh, yes, if I can sit down.
Of course.
Question. So earlier on you showed a number of slides where ultimately you showed the book lung and the gills.
Yeah.
My understanding was what you're saying, that these features have to be on the surface of the fossil for you to examine, etc., etc. And yet those images looked almost like they were a section through the fossil. Was that right or not? And how they produce? That's what we're doing.
Right. In this case, the shape, the orientation that the trigonotarbid was preserved in did allow that view into it. Right. They are open to the air, so they're not. Well, I mean, they are internal, but they have an opening to the air, and that's what they could actually just see. They were just really, it was just really, really good preservation.
So it was just chance?
Oh, yes.
Right.
Yeah, in this case, yeah, it was.
Thank you.
Anyone else? Yes.
Just taking it back a bit further, what about the other arachnids like harvestmen and mites and so on? Were they a branch off from this even earlier, pre-Silurian?
They would have been. Is that a balloon on the ceiling? So wasn't expecting that. So the earliest arachnids, there's a group called Tetrapulmonata, from which the spiders and this lot evolved. And that's a fairly stable group, but I think when we're looking at the earliest arthropods split into the major groups not long after the Cambrian. There is, yeah, it's still very, very uncertain. Yeah, it's still very uncertain. But the question is, did terrestrialization happen before, and there's one group that split, or did it happen many times, and we get scorpions and what have you multiple times? So we think probably a bit of both in that sense. Horseshoe crabs could be put anywhere. They are really a confusing group. And there's quite a lot of research going on with the horseshoe crab at the moment. So where do they fit? Do they fit next to scorpions? Do they fit next? It's really not an easy question to answer. Sorry.
Sorry to have asked it.
It's okay.
Hi, I've got another question that might also be a bit technical, but on that family tree, the phylogenetic tree that you showed, there were some numbers, and I wasn't sure what they meant. But you were a bit upset, I think, that trigonotarbid was down with whipped scorpions. Those numbers look to me like they might be confidence levels at the branch points, and that one only had 0.51. It seems to be the least certain of them. So it could be perhaps that the trigonotarbids are somewhere else, but that's uncertain.
I'd have to go back and look at the slide to tell you whether it's confidence levels or bootstrap values or molecular clock type information. Probably not molecular clock in this case, but it might be branch length. So yeah, yeah, I can go back and have a look, but currently. yeah, it might well be that it's least confident for putting it there, but that doesn't mean that it's any better anywhere else. That's the most confident that we have. Yeah, out of all the other options, it gets
where
in.
there's lots of doubt, I think, isn't there?
Yeah,
Because
there's a lot going on still. Yeah.
Well, I think you explained that very clearly, actually.
Okay.
So, yes, lovely talk.
Okay.
So thank you very much.
Okay. Thank you.